So we continue, today we continue to talk about the structures of the cell, and as far as I remember, last week we have talked about the endospores. I think we finished endospores, right? We talked, yes. I don't remember if we talked about the cell membrane. No, we didn't. Okay, good. Because this is something that should be fairly easy to you, since it is covered in virtually every biology course that is taught anywhere. The cell membrane is an interesting structure consisting of lipid bilayer. So you see those molecules of phospholipids here, where these circular heads represent the hydrophilic parts, okay? And they are exposed either to the cytoplasm of the cell or to the extracellular fluid. Both cytoplasm and extracellular fluid mainly consist of what liquid? Water, right? So hydrophilic heads are perfectly fine in terms of interaction with the water. On the inside, between those hydrophilic heads, you can see the tails of the molecules. And those tails are hydrophobic. So this hydrophobic internal layer of the membrane provides the actual separation between the extracellular fluid and the fluid inside of the cell. Think about this. If there would be no hydrophobic layer in between, the hydrophilic heads would simply dissolve, right? They will just become a part of the solution. Does that make sense to you? Good. So actually, so phospholipids means that they have a phosphate group here, which is polar, then glycerol, and two fatty acid tails. We talked about the structure of phospholipids when we talked about the biological macromolecules. This is more of a reminder why phospholipids possess such properties. Actually, molecules that have both hydrophilic and hydrophobic parts of the molecule are called amphiphilic. Amphi means both. Okay, so they interact both with water on one side and lipids on another. Now, why it's so important to understand the structure of the membrane? Because the structure of the membrane determines the transport, right? Main function of the cellular membrane is to selectively allow molecules to cross it. Now, which molecules, what type of molecules can easily pass across through the membrane without any assistance? Is it going to be water soluble or lipid soluble? Lipid soluble, right? Because you have a lipid layer here. It will not allow any water soluble molecules to pass through, but lipid soluble molecules can easily get across the membrane. When molecules cross the membrane without any assistance, this is called a simple diffusion. Now, diffusion is the transport along the concentration gradient. What does that mean in terms of the direction? So molecules go from what concentration to what concentration? High to low, yes. So usually represented in the form of that slide here. Okay, from high to low. Does this movement require any energy expenditures? No. So diffusion is not actually, in fact, if you think about it, diffusion may produce energy. Think about the energy that this ball will acquire at the end of the slide, right? It will roll with a certain speed, having a certain momentum. Now, what we're going to do with the large, or not, well, what are we going to do with the polar molecules, the hydrophilic one? How can they pass through the membrane? I mean, we are not David Copperfield. We cannot pass through the wall. What do we build in the wall? Huh? Doors, yes, we build doors. So those membranes have quote-unquote doors, the transmembrane proteins. We call them channels, carriers, sometimes pumps. These transmembrane proteins, proteins that penetrate the whole span of the lipid bilayer, they facilitate the movement of molecules, the hydrophilic molecules, from one side of the cell to another. What determines the direction of movement? Why molecules will go from, say, from concentration, yes, the concentration gradient. Does that make sense? So just even though molecules have to go through the channel, okay, they will still go from high concentration to low concentration. Does that make sense to you? Now, what if somehow these concentration numbers will flip and the concentration will become high here and low here? Will they go in the opposite direction through this channel? Yeah, sure, absolutely. Channel only facilitates the diffusion. Does that make sense? Channel does not determine the direction. It only facilitates the diffusion, like a door. Having a door doesn't mean that you can go only one way. You can go both ways. Make sense? Now this diffusion is always the movement when you don't use energy. When do you need the energy? When you move your molecules in terms of the gradient, how you move them? From low to high, against the concentration gradient. Does that make sense? So when you need to pump molecules against the concentration gradient from low to high concentration, this is called active transport. Does this process require energy? That is true, and this energy is used by pumps. You can see the example of a sodium-potassium pump, which I don't want to get into too much details about. Pretty much the idea is that every pump has a part that breaks down ATP, right? So I have ATP here, and it is broken down into ADP and inorganic phosphate. That breakdown yields energy, which is shown here as the yellow star. And this yellow star energy facilitates movement of, in this case, sodium against the concentration gradient. Does that make sense? So please understand the differences between diffusion and active transport, and difference between the simple diffusion, which is illustrated here, movement directly across the membrane, and facilitated diffusion, movement through using transmembrane proteins. Make sense? Any questions? Yes. This is simple, yes. So if you're as old as I and ever watched David Copperfield's show, he walked through the Great Wall of China directly. Well, obviously it was a trick, but nevertheless. So simple diffusion is walking through the Great Wall of China, walking directly through the membrane. Does that make sense? Good. Now, membranes, the parts of every microbial cell, in each and every one, there's no cell without a membrane. On the other hand, cell wall is the part of the cell that may or may not be present. It often is. I would say in most microbes, in a vast majority of microbes, cell wall is present. And it's a fascinating structure, really. So it consists of the copolymer. I'm going to write this word, copolymer, which means that copolymer means that it consists of two distinct types, 2, 3, 4, in this case 2 distinct types of elementary building blocks. Those building blocks are carbohydrates and peptides, right? So you can see on this illustration the carbohydrates shown as large blue and beige spheres, okay? So these two molecules, N-acetylglucosamine and N-acetylmuramic acid, form long chains of polysaccharides. Does that make sense to you? These long chains of polysaccharides are connected to each other by the oligopeptides. What does that mean? Oligo, what does that mean? The word oligo. Huh? Ever heard about the word oligarchia? Oligarchs. Not a lot. I mean, more than one, but a few. Not many. Okay? So oligo means a few. I cannot tell you, and I don't think anyone will be able to tell you, when a few ends and many begins. So like 15, is that still oligo or it already is many? Poly. Okay? So you can see that the number, so each oligopeptide, this linkage, oligopeptide linkage, each of these linkages consist of several amino acids. Make sense? So you have it's copolymer of carbohydrates, monosaccharides, and amino acids. Okay? Does that make sense to you? Now, if you think about the structure, and we'll talk about gram-negative, it's not very much different. But think about this. Look at, you have clothing on you. Okay, look at the fabric. If you look at the fabric, it's a lattice. Okay? You have threads going in one direction and threads going in a perpendicular direction to the first ones. Same can be observed in the wall. You have threads going in one direction, right, that are connected by short linkages in an opposite direction. You essentially have a lattice that is really, really tight and rigid and has a great mechanical resilience. Does that make sense? Now, gram-negative cell wall structure is just a little different because this oligopeptide linkages are slightly shorter. That's it. Other than that, both gram-negative and gram-positive microbes have fairly similar chemical structure of the cell wall. However, the overall structure of the envelope that surrounds the cell is very much different between gram-negative and gram-positive. And we're going to talk about it just right now. And gram stain actually reflects that difference. Okay? We'll get back to peptidoglycans. Let's talk about the structure of the cell walls, first of all. So gram-positive cells, they have very thick layer of, that's a membrane, that's very thick layer of peptidoglycan, okay? So you have a membrane here, let me color it in a different, okay? A membrane, and you have a cell wall. Does that make sense? So they have two layers. Gram-positive cells have two layers. Membrane and very, very, very thick cell wall. Gram-positives like Staphylococcus aureus. In the cell wall, in that layer of peptidoglycan, you're going to find so-called teichoic acid. You see those sticks here? Okay, so teichoic acid plays many roles. It's teichoic acid is a unique component of gram-positive cells. Okay? Only gram-positive. Okay, it regulates the rigidity of the cell wall. It may help with the adhesion to the surfaces. How can it be a virulence factor for cells? Adhesion. Yeah, it sticks better to the cells, right? If it sticks better to the cells, that's your virulence factor. Does that make sense? It even helps with the transport across the wall. Gram-positive cell wall. Is that clear? More or less. Clear, okay? Two layers. Membrane and thick peptidoglycan, okay? Think about it as a sandwich. So I have a thin layer of bread here. Okay, that's bread. And that's your cheese, bacon, turkey, whatever you want to put in the sandwich. No bread on top. Just at the bottom. Okay, what about gram-negative then? Well, in this case, you have a membrane. Again, cell membrane is present everywhere. And then you have fairly thin peptidoglycan wall. Make sense? And then again, you have another membrane, so-called outer membrane. So outer membrane is also a phospholipid bilayer, but it incorporates several important other important components, okay? For instance, murein, the special protein, attaches the external membrane, the outer membrane, to the cell wall. Porins, special proteins, serve as channels. They allow transport of chemicals across the outer membrane of gram-negatives. And last but not least, let me erase it. You see this long red molecules? This is lipopolysaccharide, or LPS. This is it's a distinctive feature of gram-negative microbes. It is a bacterial toxin, which means if this toxin gets in the circulation, it causes fever, very high fever. Okay, so that's why if you have bacteremia, microbes in the blood, and even if those microbes are dead, lipopolysaccharide will be recognized by your immune cells, will be recognized by your hypothalamus. It will produce immune response, which may actually be very, very bad. I mean, very strong, bad for you, like a toxic shock. And it also, when LPS is recognized by hypothalamus, it produces the fever. Okay? Yeah, okay. Now, let's go back. So, do you understand the basic concept? Let me summarize it for you. So, gram-positive, membrane and thick cell wall. Gram-negatives, membrane, thin cell wall, outer membrane. Gram-positives have teichoic acid in the wall, helps attachment, assists in transport. Gram-negatives have porin proteins in the outer membrane that assist with the transport, and have lipopolysaccharide, which serves as an important, very, very important virulence component of the cells. Are we good? Yes. So again? Yes. Yeah. Not really. That's interesting. It's a good question. It turns out that LPS does not cause antibody formation. So your immune system is not gonna form antibodies, and it's a unique, sort of a unique thing. Okay. This is also, bless you. This is also called endotoxin. Endo means from the inside, right? So it's endotoxin. It's a component of the cell, and it does not cause the formation of the antibodies. And I'll talk to you about it. We'll get to LPS in a second, okay? One thing that I want to highlight is that I just realized that I made this understatement. Some antibacterial drugs interfere with the cell wall synthesis. Drugs like penicillin, everybody heard of penicillin in this room, I suppose, and vancomycin, they prevent cell wall from being linked, essentially. So this linkages, peptide linkages, are gone. How do you think it will change the cell wall properties? It will become weak, right? And bacteria will just explode. Now, which bacteria will be more sensitive to penicillin? Gram-positives or gram-negatives? Why? But it's protected by the outer membrane from the outside, so penicillin has harder times getting there. Gram-positives. If you break down cell wall, there's nothing. It's just a membrane, so it just pops. Okay, and if you would look at what penicillin and vancomycin used for, when you go with a sore throat, one of the first choice is penicillin or its derivatives or other beta-lactam antibiotics, okay, that interfere with the cell wall synthesis. That makes sense? Now, a few more words about lipopolysaccharide. So lipopolysaccharide consists of three major components: the O antigen, that's different between different species, polysaccharide core, and the lipid A. Now, why I pay so much attention to LPS? Imagine that you have an isotonic solution that you inject to a patient. Imagine. So do the IV injection. An isotonic solution is essentially water and salt, sodium chloride. So you can mix them all together, put them in the bottle, stick the bottle in an autoclave. Autoclave it, kill everything, and then inject it into the patient. Right? Right? Wrong. If you had microbes in that mixture, when you autoclave, you kill the microbes. Cells are destroyed. However, this molecule, LPS, a bacterial endotoxin, doesn't go anywhere. It stays in the fluid. So when you inject it to a patient, endotoxin works as the pyrogen. Patient develops a fever. So for this, it's a little bit, in this course, I give you way more than I'm going to ask. So don't worry, I will tell you. What I'm going to tell you is FYI part of the course. I'm not going to ask this particular story on the exam. It's not in the PowerPoint. There's a method. First of all, to get rid of LPS. Second, to test if it's present. To get rid of it, we're not autoclaving, we filter solutions. Filter with a pore diameter of 0.1 nanometer, which is very, very little. Very little. Bacteria do not cross it, so there's zero chance that endotoxin, LPS, will cross the barrier. Does that make sense? What if we don't know if our solution contains or doesn't contain LPS? What if we're not sure? Can we test? Yes. Turns out, have you seen horseshoe crab? Horseshoe crabs, they don't have blood, they have a fluid called hemolymph. it's more primitive than human or any other mammalian blood. It contains, instead of red blood cells and platelets, it contains cells that are called amoebocytes. And the immune response that they produce when they detect bacterial endotoxin, they form a clot, those amoebocytes. So they restrict the bacteria in a certain area of that horseshoe crab. And that was discovered. The hemolymph of horseshoe crabs was collected, amoebocytes were isolated, then they were broken down in pieces, so it was lysate, okay? And what you can do, you can mix your sample with that preparation from amoebocytes. If you have endotoxin, you will see a clot. If you don't have endotoxin, there will be no clot. It's extremely sensitive method. It goes way beyond the concentrations of endotoxin that can cause pyrogenicity. Sort of a fun fact, which state is famous for horseshoe crabs? Crab state. New England. Goddamn Patriots. Massachusetts, right? So it turns out I think every spring, thousands of horseshoe crabs migrate and they crawl on the shore of Massachusetts. Special farms catch them, bleed them, quote unquote, not to death. They just collect some of the hemolymph and release them back in the wild. And it's extremely convenient because the only other way to test for pyrogenicity is to use rabbits, inject them with whatever you're testing, and then stick the thermometer up their ass and measure temperature. And that's the task itself. So rabbits are awful animals. Now, we talked about gram-positives. We talked about gram-negatives. Are there any different types of microbes? It turns out there are acid-fast microbes, Mycobacteriaceae. If you would look at the structure of the cell envelope in mycobacteria, you will notice that they are pretty much gram-positive. They have plasma membrane, they have cell wall. All the same, right? All the same. But on the outside of that cell wall, there is a layer of mycolic acid. It's a waxy polymer it's a hydrocarbon polymer, okay? And functionally, mycolic acid protects mycobacteria from immune system. Mycobacteria, like tuberculosis, okay, Mycobacteria tuberculosis or Mycobacteria leprae, those belong to the group of Mycobacteriaceae. And you can detect the specific type of microbes using acid-fast stain. You can see acid-fast bacteria here in this micrograph. So they're going to appear red under this stain. Some bacteria, like Mycoplasma, they don't have cell wall at all. They have a membrane, but not the wall. So they don't have a very distinctive shape, obviously, because they can change the shape. Does that make sense to you? They're also pretty sensitive. Mycoplasma is an interesting pathogen. We're going to talk about it. It causes walking pneumonia. Can cause walking pneumonia. They're called mealy-cutes. I really like the cute part. Well, mealy-cute says that it's a microbe without rigid cell wall. Firmicutes, microbes with cell wall. Okay? A Mycoplasma is not only the microbe that does not have a cell wall, it also is the smallest independently living organism on Earth. Mycoplasma has something like 500 or 530 genes. It's really tiny. You cannot see it with the light microscope. The best chance is to use fluorescent. You can stain mycoplasma specifically and detect it in the cells. It's usually an intracellular pathogen. What about archaea? Remember, I mentioned that archaea are different from bacteria. Well, quite different. They don't have peptidoglycan. They have similar polymer, but not peptidoglycan, and some have. So when we say glycoprotein, it means that they have more amino acids than carbohydrates. Some of them don't even have amino acids. The cell wall is composed entirely of polysaccharides. So take-home message. You have to know the distinctive features of gram-positive, gram-negative cell wall, why Mycobacteriaceae are different, what makes them different, the mycolic acid on the outside. Understand that some microbes don't have a cell wall, and know the functions of the parts of the cell envelopes, such as teichoic acid, lipopolysaccharide, and porin. Lipopolysaccharide and porin for gram-negatives, teichoic acid for gram-positives. And of course, ID of peptidoglycan. So I will not ask you the names. Don't you worry about that. Okay, don't worry. But I may ask you, which two basic biological molecules make up the cell wall? And you have to say it's carbohydrates and amino acids. Make sense? I hope I made myself pretty clear. Now we go into more and more outside of the cell. We're finished with the envelope. Almost. The last thing to talk about here is the glycocalyx. It's a layer, usually of carbohydrates, that surrounds the cell from the outside. And it's not present in all the cells. Okay? So it may have two distinct forms, the form of capsule and the form of slime layer. Again, I brought this example before. When you walk on the rocks in the pond or in the lake or in the river, they're slimy. Microbes that form that slime actually have the slime layer outside their cells. So slime layer and capsule are different mostly in terms of the appearance. Capsule is more rigid. Slime layer is more loose. You see the difference? Actually, if you would look at the colonies of microbes that have capsule, you will see that they have mucoid appearance. Mucoid, in very unpleasant, easy terms, looks like it's somebody's snot. Okay? It's not clear, it's like mucus. Okay? You can actually say, you can see the capsule or slime layer or judged by the colony appearance. Okay? So what is the function? For both of them, they help to adhere to surfaces, both of them. Okay? So that adherence to surfaces help microbes to form biofilms. Capsule is also instrumental to protect cells from phagocytosis. So if cell is encapsulated and macrophage engulfs it, cell may survive inside of the macrophage and not being killed, which can be counted as the virulence factor. Does that make sense? We will talk, when we will discuss microbial genetics, we will talk about Griffith experiment, when he was studying 2 strains of Staphylococcus aureus. Wait, no, Streptococcus pyogenes, sorry. 2 strains of Streptococcus. One had capsule and one didn't. Well, guess what? The one with a capsule killed mice. The one without capsule didn't. See the difference in pathogenicity? That's what capsule can do. So, protection against phagocytosis, 1. Second, formation of biofilms. What is the biofilm? Biofilm is the community where microbes live in the mutualistic relationships or synergistic. Can anyone define what is synergistic relationship? Not only working together, there's some consequences of working together. Huh? Oh no, they don't. When they're in the synergistic, they don't compete. What is synergy? Think about this. You have 2 microbes. Each of them can live independently. Not a problem. Make sense? But when they live together, they become way more successful in terms of reproduction, each one of them. Okay? So synergistic relationship it's not mandatory. They can live without it. But if they are in those relationships, if they are parts of the community, each of the microbes will benefit from that relationship. It's like a soccer team. Okay? I mean, each player can play by himself, but only when they are the team they can win. Okay? Mutualistic is slightly different. Mutualistic relationships are mandatory. Microbes cannot survive otherwise. And when relationships are mutualistic, it means that both microbes, or 2 or 3 or 5 or 25, they benefit from that relationship. Right? So, in biofilms, all members of the community benefit from being a part of its community. Why biofilms are so important? First of all, they help to retain water, and drying is one of the major enemies of microbes. They much better in avoiding phagocytes. And antimicrobial agents have tough times to penetrate the biofilm. If you will talk to orthopedic surgeons, they will tell you that when they place prosthetic joints in patients, patients often have to be on the antibiotics to the rest of their lives. The reason for that is it's really hard to make the surface of the joint, the prosthesis, completely clean. So there will be some microbial cells. When they end up in the joint, they will start to reproduce. And antibiotic therapy can keep them under control at least. Because if they form the biofilm on the surface of the joint, it causes severe inflammation around that implant, right? And it may require actually cutting that out and cleaning this whole thing up and replacing it again. So biofilms is the big problem. Catheters. On the surface of catheters, biofilms form very quickly, and it facilitates the spread of the bacteria, say, up through the urinary tract. Okay, and if they spread, it's really hard to. I mean, you can control microbial populations in the organs, but antibiotics will not penetrate through the to the catheter. Yes. If I'm not familiar with the biofilm formation on IUD, but I can imagine microbes setting up the biofilm, and I believe it may be problematic. It may cause problems because it's technically part of the body and it will be exposed to antibacterial agents, drugs, right? But drugs have such a hard time killing the biofilms. Turns out, like, boiling may not necessarily kill the biofilms. Okay, we talk about hydrogen peroxide exposure for 10, 20, I don't know, 30 minutes, an hour to kill all of them. And I believe nobody's going to do that to humans, you know, pour hydrogen peroxide inside of the body. So it's really a problem, okay? That makes sense? Now, in some microbes, there's an S layer. We don't know what it does. It's a mixture of structural proteins, glycoproteins on the outside. Maybe it's a cell wall. Maybe they help cell to retain rigidity and not being blown up by osmotic pressure. You can see the capsule here. You know this white structures around the cell? That's the capsule. Probably around Staphylococcus. Does that make sense? Capsule and the slime layer. Know the difference. Know what they're made of. Know the function. Difference is pretty simple. Okay. Flagellum. Well, external appendages. So we're going to talk about 3 flagellum, fimbriae, and pili. And normally this picture can scare the living daylights out of anyone, including me. Don't worry about the structure of that stuff here. Okay? Don't worry. The idea that I want you to understand is that flagellum is a rotating thing. There is a motor inside of the cell. So this is going to be your motor. There is a hook that goes through the cell envelope, through the membrane, through the wall, and stuff like that. Okay? And there's a filament right here. Filament actually propels the cell forward. Does that make sense? Motor rotates the hook. Does that make sense? So hook goes like that. And filament follows it and actually becomes a propeller. Okay? Sure. Awesome. So this is the hook. Imagine that there's a filament attached here. Okay? My hands are the motor. It rotates and propels the cell forward. Does that make sense? What does this process require? Energy. Sure enough. So ATP. Okay? Now, here's the funny thing. If you remember oxidative phosphorylation, if you remember the aerobic respiration in the cell, you may remember that the very last step is when protons, hydrogen ions, diffuse through the membrane, and the energy of diffusion is converted into ATP. In this case, ATP creates the hydrogen ion gradient, the proton gradient. And when protons diffuse through certain channels, they fuel up the motor. There was actually that movement of protons was measured. It's like flagellum requires something like 80 hydrogen ions. They actually counted each and every one. I don't know how, don't ask me. Okay? So this flow of hydrogen propels the flagellum, okay, and flagellum propels the bacteria forward. Structure is slightly different, okay, between gram-positive and gram-negative. So this is going to be your gram-positive, this is going to be your gram-negative. That's the statement that I want you to know. Structure of the flagellum is different between gram-positive and gram-negative. That's it. Okay? Now, flagella can be arranged in a different way. This is you have to know. Flagellum can be monotrichous, one flagellum at one end, amphitrichous, two flagella on the opposite ends of the cell, Lophotrichus, a bunch of flagella at one end, and Peritrichus, flagella all over the surface of the cell. Does that make sense? It actually makes a lot. So mono, 1, amphi, 2, peri, all over the place, lopho, bunch. Right? Other appendages. Fimbriae. Very short hair-like extensions on the cell wall that helps with adhesion. Okay? So again, it's a virulence factor because they help with adhesion. Cells that have fimbriae will stick to the cells better, will be more likely to produce the infection. Pili. Pili are longer appendages. You can see them here. That made of a protein, they're like tubes, okay? And special types of pili called sex pili allow microbes to exchange the genetic material. You can see two bacterial cells connected. So this is one, this is another. They are connected by the sex pili, okay? This picture, they are connected by the sex pili that allows them to exchange DNA. Remember we talked about the cell division, and we chatted a little that bacteria do not have sexual reproduction. And sexual reproduction, say in mammals, is the major contributor to genetic variation. Right? My mom and my dad are very different, so I'm something in the middle. That combination of genes creates the genetic diversity. If bacteria divide, every daughter cell is the same as the mother cell. Mutations, well, yes, mutations, but it's not that fast. We would want something more profound. So this comes from pili, from exchange of genetic material. Actually, this process, when bacteria are connected by sex pili, is called properly conjugation, but microbiologists usually call it a bacterial sex. Okay? Why it's so important? Because of this. Microbes can exchange genes that contribute to virulence, and what is most important, to antibiotic resistance. It's pretty obvious that if you have E. coli that is resistant and E. coli that is not, two cells of the same species can quite easily exchange genes. It's just obvious. Turns out, different species can also exchange genes. So that's why use of antibiotics it should be limited. We'll talk about it, but in the last 2 visits, my wife visited doctors 2 times in the last maybe 6 months with a respiratory infection. They did everything not to avoid antibiotic prescriptions. And on one hand, I completely understand them, but on the other hand, I'm totally mad. Because she suffered for extra few days without antibiotics. Any questions on the appendages? Did I make clear what you have to know? No function of flagella, no function of fimbriae, no function of pili. Okay, flagellar movement, fimbriae attachment, pili genetic exchange. Now, movement by flagellum. I like the name. It's run and tumble. It's called run and tumble. So, when bacteria runs, flagellum moves into the counterclockwise fashion, and bacteria follows a certain cue. The cue is chemical gradient, which is called positive chemotaxis. Okay? Usually it's the presence of nutrients, so bacteria move towards the nutrients. Make sense? Good? When it tumbles, flagella move in the clockwise direction, and that helps the bacteria to orient itself along the gradient of attractant. Does that make sense? No? Not really? Imagine a bunch of guys that come to the state fair and they just run around trying to find where's the beer. And then, ho, the beer, and they all run towards the beer. Okay? So that's like bacteria, okay? They try to figure out where is the nutrient, the chemical, whatever they look for, where it is abundant. So they kind of run in place trying to figure out the orientation of that gradient. And then, we need to go that way because concentration of the chemical increases towards that way. So they orient themselves and they move along the gradient. So when they orient themselves, they tumble. When they finally know where to go and they move towards that chemoattractant, move to when they follow concentration, exhibit positive chemotaxis, that's run. It does not have to be chemical. Yes? Yes, yes. It doesn't have to be chemical. It can be a magnetic field. There are magnetotactic bacteria. So they will figure out what are the lines of magnetic field. When they orient themselves, they're going to move towards the magnet. They're going to swim towards the magnet. Okay? There are phototactic bacteria that will move towards the light. That makes sense? So in this case, chemo, chemical, taxis, movement. Alright? Make sense?